I wanted to discuss the issues associated with building maintenance, water damage, and its impact on the occupants of these indoor spaces. In order to appreciate it, we have to sort of have a sense that this is a cyclic process. Let's assume that there is a roof leak. It's going to leak when there are storms, and then after the leak has occurred, water intrusions moisten building materials, and then within 24 hours, various species of molds will start to grow. These are predominantly species that favor growth in higher humidity and dampness than many of the ambient air species do. And so we are able to differentiate the history of a building if we know what the nature of and the distribution of the species is. And we do a sampling of spores by just taking a dust sample 8 inches away from the wall on the surface of the floor. We will basically have spores that represent the history of the building. And the reason that this is the case is because spores are microscopic. They range in size from perhaps as small as a half micron, but usually between 1 and 7 micron in diameter. And these particles move around the house or the building with the slightest movements of air. The other particles that are present that also represent a significant hazard in the bioaerosols that are associated with damp indoor environments are the cell wall fragments coming off of the desiccated colonies. The reason that they represent a significant hazard is because on the surface of these submicron sized particles are to be found the digestive enzymes that are secreted by the colonies during the course of their life cycle, and the toxins that are secreted also through the hyphae, or these small microscopic tubules, which is the way that the colony delivers its digestive enzymes and the poisons that it's capable of making, if it is a poison-producing species, onto the surface of the colony. And then when the colony dries, they dry on the surface of the cell wall fragments. So now, in the sort of ecosystem of repetitive intrusions of water, the spores and these particles that are left behind actually gradually disperse throughout the building. Every time water intrusions occur, from plumbing leaks, from window leaks, from roof leaks, from improperly caulked foundations, whenever the water appears, the colonies start growing again. And this cyclic process is the root of the generation of particulate matter in the size range of the particles in smoke. And these particles do indeed move around the entire building, even if there is just one source of water intrusion. It is that movement with the slightest movement of air that again disperses these particles throughout the entire space. In the process, creates the circumstance in which occupants get exposed. When we walk, we raise the dust. During the times when a building is inactive and there's nobody in the building, the dust settles down to the floor again, waiting for another period of activity. And with additional activity and the entrance of people into the living space, the working space, or the study space of whatever the building is, these particles are raised. The problem is we don't see them. So because we don't see them, our awareness of them is clearly not prominent. But the particles are really prominent, and they really create a major issue, because once they are raised into the air to the level of our nose and our mouth, we will inhale the particles and the poisons on their surface, whether they're poisons or digestive enzymes. The matter of digestive enzymes is another dimension of this conundrum and problem. The species of fungal organisms that we're concerned about and the overall kingdom of fungi acquire nutrients in a manner that is distinctly different from animal cells, which is the other reservoir of what are called eukaryotic cells. Eukaryotes are species that cannot fix energy from the sun. All of our plant matter has the ability to generate oxygen from carbon dioxide through photosynthesis. In the absence of photosynthesis, a species becomes entirely dependent on other organic material as its primary source of nutrients and energy, in a process that is different than the animal cells. Animal cells engulf a particle that is nutrient material by wrapping the membrane of the cell around that particle in a process called phagocytosis, and then they bring that little sack of material into the interior of the cell, where there is another packet of digestive enzymes in an organelle called the lysosome. These two marry together, and the enzymes in the lysosome will basically break down protein to amino acids, carbohydrates to simple sugars, fats to triglycerides, and DNA to DNA bases. And now you have a reservoir of building blocks and nutrients inside this little sac, and the cell basically uses it and consumes it for purposes of growth and energy. In the case of fungi, they actively secrete their digestive enzymes onto the surface of the colony. And once they have done that, all of the subsequent particles that are generated from the surface membranes of the colony will have digestive enzymes on their surface. And of course, although there are hundreds of thousands of species of fungus and mold, and only a handful, maybe a few hundred, are capable of producing poisons, those species that can produce poisons will be producing those poisons from the moment that the spore first propagates a new organism. They may vary in the amount that they secrete based on their own nutritional status, but David Strauss from Texas Tech, the Department of Microbiology, and his group of researchers more than a decade ago showed us that any species capable of producing toxins will do so from the moment they begin to grow. So if there are Stachybotrys spores present, as an example of one species capable of producing extreme toxins, those toxins will be present in the environment that the organisms are growing in from the moment they start to grow. And of course, that represents a significant problem, and it creates a situation in which the particles themselves are extremely antigenically active, meaning they will stimulate an immune response in whatever organism they find their way into, because to the organism that they are now in, they are a foreign protein. And the immune system's mission in your lifetime is to intercept any organism that is producing non-self protein structures and destroy it. Because to the immune system, that's the signaling that there is an infection about to occur. And as a result of that, when these particles coated with digestive enzymes and poisons reach the alveoli of the lungs, the presence in the lungs and in the air sacs and smallest airways of these inflammagens implies that there will indeed be inflammation, mobilization of the immune cells of the lung in order to fight a war, because it's defending the lung against what it is perceiving as a foreign invader, whether the invader is growing or not. And of course, with spores, being the seeds of new life, it is only appropriate for the immune system to try to destroy them or to try to deal with them in some way that eliminates them as a potential infectious threat. So, in water-damaged buildings, we have the conditions because of the presence of cellulose, because of the presence of the spores from fungi and also bacterial forms that grow when water is abundant. We have an entire ecosystem that's present in these indoor structures, and it is abnormal. The primary and most serious route of entry for the poisons associated with fungal organisms is the respiratory route. There is a publication called The Medical Aspects of Chemical and Biological Warfare. This treatise was released by the Surgeon General of the United States in 1996 or 1997. In that textbook, which is a 2-volume text, there is a chapter on the poisons that are produced by the Stachybotrys mold species. Those poisons are referred to as trichothecenes. Trichothecenes is a class of compounds known as epoxides. At least in that article, they suggest that there are 149 different epoxides. And these compounds share the chemical properties of epoxides, which is that they have very, very high reaction coefficients and they tend to bind to things very intensely. So once they have gotten on the surface, let's say, of the colony or the particles of cell walls or clothing, furniture, electronic equipment, once they're present, they're extremely difficult, if not impossible, to get off. And as a result of that, that circumstance of having the toxins and the spores from Stachybotrys in particular represents a higher level of hazard and a much greater problem in terms of remediation than most of the other species. The implication of that, again, is that you cannot safely take things from one indoor space that has been contaminated with Stachybotrys and a species known as Chaetomium, both of which have spores that tolerate extreme conditions and are still viable. According to Chapter 34 out of the textbook on the medical and biological aspects of chemical warfare, the most toxic route of exposure for particulates with trichothecenes present is the respiratory route. If you inhale this material, you will have a 10 to 100 times greater level of toxicity than if it's injected intravenously. Of course, the question arises, how is it possible that a poison administered by inhalation is worse than having the poison injected in your bloodstream? And the answer lies in the unique physiology and biochemistry of the lungs are an amalgamation of very tiny microscopic respiratory surfaces that we call alveoli or air sacs. The alveoli average 10 micron in diameter, and in order for them to function properly, they must have a moist surface in order for the gases in air to dissolve and then cross into the capillary bed and be distributed throughout the body, so that oxygen exchange and carbon dioxide elimination must occur in a moist surface. The properties of water are such that the water molecules like to attract each other. And this is most evident when you have a little dropper and you squeeze the beginning of a drop out of the dropper. It hangs in place. And the reason the drop hangs in place is because the water molecules individually have attractive force for each other. That attractive force is called surface tension. And the problem for the lung is that if we don't have something to help reduce the surface tension of the water molecules, the air sacs in the lungs will collapse and stick together. This problem looms very intensely in premature babies. The compound known as pulmonary surfactant, which is required in order to lower the surface tension of the water and allow the water molecules not to attract each other as intensely as they normally do, does not develop in the fetus until after the 27th week of gestation. And if a baby is born before that time, there is a very high risk that their lungs are not going to be able to remain open. And as they collapse, eventually the baby dies of respiratory distress syndrome. So we have very elaborate neonatal intensive care units to overcome these problems. And in some cases now they're actually aerosolizing the pulmonary surfactant. So what is this pulmonary surfactant and why does it have the effects that it has, and how does that affect the toxicity of mycotoxins or any other toxin that's present on the surface of respirable particles that end up in the air sacs? Pulmonary surfactant is basically a detergent. If you remember, detergents are used in water to do our laundry so that we can lift the oils off of the cloth, so that we can get the grease and the grime off of the clothing and the cloth material and have it go off and be rinsed away after it's released from the fabric. Detergents are the things that do that indirectly, because what they do is they create a circumstance in which the water molecules are not attracting to each other as intensely, and they can then get in, around, and under the molecules of oil and literally make them dissolve in water, which they don't normally dissolve in. And then once having dissolved in the water, it is very easy for them to cross over into the capillary circulatory bed and enter the bloodstream. And in fact, when they do enter the bloodstream after being washed off the particles in the lung, having been made miscible or mixable with water, they are able to achieve higher concentrations and disperse more intensely throughout the body through the bloodstream than if the material was injected directly into the bloodstream, because if it were to be directly injected and it were insoluble, it will form little droplets, just like fat circulating in our body circulates in clumps or spherical droplets. That means that less of the actual chemical is available to interact with the surfaces of the walls of the vessels and the other tissue than if it has been made miscible, the same poison, in the lung as a result of the pulmonary surfactant. So this appears to be the mechanism through which greater amounts of these toxins enter the circulatory system and get to tissues, and that is why the LD50, which is the lethal dose 50, or the dose that will kill 50% of the test animals or organisms being studied, is 10 to 100 times lower when these particulates with the poisons are inhaled versus other routes of entry. So now, with a little bit of an understanding of the unique circumstances in damp indoor environments, we can begin to appreciate the level of toxicity that is being seen among the occupants of these buildings.